5. Cattle with bovine spongiform encephalopathy, BSE, have microscopic holes in their brain tissue. This disease involves the misfolding of proteins which then form clumps. Clumps of misfolded proteins can be seen with an electron microscope. BSE is diagnosed by examination of brain tissue. Explain why the clumps of misfolded proteins can be seen with an electron microscope but not with a light microscope. So, once again, Edexcel will always bring you a disease or something that you probably haven't heard of. You probably would have heard of BSC, maybe you've heard of mad cow's disease. And even if you don't, because it's not in the specification, it's not part of the specification or in textbooks, you wouldn't have come across it or studied it. You may have heard of it, but you don't need to know any of these background diseases or anything in order to answer the questions, but they at Edexcel have a habit of always putting in these types of questions where they will give you something that you haven't heard of and then give you a question which you can answer because it is part of the specification, so you would have come across it, but it seems like you can't, or it puts you off. And so here when they say why can you study it under the electron microscope and not a light microscope? Well, you've never heard of clumps of misfolded proteins, and so you go, well, I don't know why you can see the misfolded proteins. But the questions actually come for your knowledge of the microscopes. And so when you've come across microscopes, you would have learnt about magnification and resolution. And you would have known that electron microscope has a better resolution and has a better magnification than light microscope. And that's the answer. That's it. That's why you can see the clumps with the electron microscope but not with a light microscope, because the electron microscope has better magnification. By the way, it's not enough just to say it has better magnification. You need to specify that you're saying the electron microscope has better magnification, because that's an easy way to lose marks by saying better magnification or it has better magnification. You're always supposed to say electron microscope has better magnification, or a light microscope does not have limited magnification. And the other mark we'll also be going for, well, the electron microscope has better resolution. And that's it. And there's another answer that you can add, and it's because the wavelength of visible light is more than a beam of electrons. And that's basically describing both how these microscopes work, whereas electron microscope uses a beam of electrons and the light microscope uses the wavelength of light. But these points are enough. Explain why stains are used when preparing tissue for examination using a light microscope. So once again, it's just testing your knowledge of microscopes and how we prepare slides, and in this case, while we're using stains. Well, you review stains in your lab practicals, and stains provide better contrast. And what that means is that stains will attach to specific parts of the cell. So stains are taken up by specific parts of a cell. Of the cell. And so what that means is that stains can be taken up by things that are inside the cell that won't always show if maybe the clumps of protein are present. So for example, you get brain tissue and one has the misfolded clumps and one doesn't have, that has the normal proteins. And so when you add the stain, if the stains are taken up by the clump, then they will show as dark, maybe blobs on the slide. Whereas a normal tissue won't take up the stain, and so you'll have these dark points or blobs on this stain on the cell, and that's why it provides better contrast. So you would have worked with stains, and once you've put something under the microscope with a stain, you'd have seen that there are specific parts that show up more, and that's because specific stains are taken up by specific parts of the cell, and that's due to the presence or absence of certain elements that you're looking for. So in this case, it's the clumps. So, the misfolded proteins have a changed secondary structure. Therefore, they cannot be digested by some protease enzymes. Describe what is meant by the secondary structure of a protein. Okay, so straightforward question. Secondary structure of a protein. It's the folding of the polypeptide chain. That's the first point. Folding of polypeptide chain. The second mark is that it folds into an alpha helix or a beta pleated sheet. So alpha helix or a beta pleated sheet. And another mark you can put in is that you will have the presence or the formation of hydrogen bonds. So after this occurs, in the secondary structure, you have formation of hydrogen bonds. Once again, just because it's 2 marks, you don't, and you have more than 2 points, you can list all 3 of them, because you will never lose marks for adding more information. Explain why the misfolded proteins cannot be digested by some protease enzymes. Again, testing our knowledge of enzymes and the shape of enzymes and the active site. And it's always the same answers, but the questions are always phrased differently. So the more past papers you do, the more that you realise that all the answers are the same. It's just a question, and how they ask the question changes. And once you realise that, you are on your way to getting that A grade. So, let's start off. Why can it not be digested by protease enzymes? Well, we've said that protease enzymes have specific active site that's specific to certain amino acids in a protein. And so you can say active site is specific to certain amino acids in the protein. And so, well, whenever there's an explain question, you always think of why. So always say why. So why is active site specific to certain amino acids in a protein? Why is that important? And so that's because the misfolded protein will not fit into the active site. And so protein not fit into active site. Well, why does that mean it can't be digested? Well, because it's unable to hydrolyze the peptide bond. Peptide bonds are present in proteins. It's worthwhile whenever they give you an enzyme question and they give you a specific substance. If it's protein, then mention peptide bond. If it's a carbohydrate, then mention glycosidic bonds. If it's a triglyceride, then you can mention ester bonds. And if it's a DNA, then you can mention phosphodiester bonds. So always see where you can fit in your extra knowledge, because that's what they're looking for. And that's it.